functions, and then a little bit more about BaseSpace. Okay, and then also I have a short presentation about what to look at, how to look at the data quality. Good. So this is the workflow. The primary analysis, I mentioned it yesterday, it happens directly on the instrument. So this is similar to all other systems like MySeq. It is managed by the NextSeq control software, NCS, and by real-time analysis software, RTA. So they are both running automatically. You don't really need to care about this. And the output is BCL files. Yeah? So it's not FastQ like in MySeq, because MySeq then has another piece of software. This is MySeq Reporter. And the capacity of this software is not enough for the data from NextSeq. Yeah? So this is why we have to run this separately. And this is then running either in BaseSpace in the cloud or in BaseSpace OnSite, or you use the BCL to FastQ converter version 2. And so this software you can just download from our website, but you need to install it on a Linux system. So that's the so that's the alternative. Yeah, but of course you have BaseSpace OnSite, so you should do it this way. And then finally, visualization and secondary analysis of data. Yes, this is what I just said. So the output data, from NCS, of course, first you get images, intensities, base calling. So this you all get directly on the machine, and then the alignment and variant detection. This is what happens in BaseSpace, either in the cloud or on site. So this is basically what you are used to from the MySeq. So at the beginning of the experiment, you generate this hierarchy of folders. So the big subfolder is the run folder where you find everything. And then after, in the first 5 cycles, we generate the cluster template. And then in the first 25 cycles, we calculate the normalization matrix and a matrix that corrects for phasing and pre-phasing. And then we can do the intensity extraction and correction, and then also the base calling. So after 25 cycles, you can also see the first quality data. And so this is common for our systems, and this is why you should not, even if you work with microRNA, you should not make reads that are smaller than 25. So 26 is kind of minimum. I think you know the structure. So the thumbnail images, this is good for quality control. Unfortunately, I just saw the quality is not so nice. Yeah. So we have to see exactly what happened, but we have some warning about the focus. So also it would be interesting to see images. Yeah. So this is what we can use for troubleshooting. This is, I think, not new. New is that we have only the 2 channels, green and red. So you have the image data and the run info XML with the specifications for this run, and then the the output is intensity data and logs. These are the locations of individual clusters. And then we detect the cluster position. Yeah, I said, yeah, the clusters are bigger on the NextSeq, so it's not 1,000 copies. We have here 5,000. And 2 micron, yeah, with other systems it's smaller than 1 micron. Because the resolution is not so high, but the surface of the flow cell is much bigger. So this is how we detect the clusters. And since we can have overlaps, it's good to use not only the first cycle, but the first 5 cycles. And for the NextSeq, it's important that in the first 5 cycles, you have a minimum of 1 base that is not G. For the index, it's stricter because it's shorter. There, the first 2 bases cannot be both a G. Yeah? So one has to be at least something else. And this is why this one index 705 does not work. Yeah? So all others work for the NextSeq, but this one you should not use. So that's the principle. Yeah. So here signal in red it's C, signal in green it's T, signal in both it's A. No signal is G. Yes. So yeah, this is the case of overlap. And then if we take the signal in the following cycle, usually we can resolve this because we have always 75% likelihood that it will be different in the next cycle. And then we have this offset, and this is due to the difference in the wave in the path length of the individual color channels. So the 2 images they are not exactly overlapping, and we need to correct for this. And this is also done in these first cycles. Yeah, so we calculate a matrix with the offset. And all these calculations, they work best if you have a library that is of high complexity. Yeah? So if you, for example, a library of low complexity is, for example, Amplicon library, where you have only a few amplicons which are amplified by PCR. So you have almost identical sequences in most of the clusters, or in many of the clusters. Maybe you are only looking for one notation, and then this normalization and offset matrix calculation does not work properly. Yeah, because you have like from one cycle to the next, you have huge differences in intensity. Normally, it should be a kind of random distribution of intensities. So this is why you should try to avoid extreme low complexity. So if you have one sample with low complexity, you mix it with other samples with higher complexity, then this can be avoided. You can spike in phi-X. You can also spike in another high complexity library, maybe whole genome or exome. So there are different ways, yeah, RNA-seq. If it's about the index read, also if you have only 2 indexes, it means that in the index read 50% of clusters are identical. Yeah, so it's better to work with more indexes when the read is of higher quality. Yes, this is about intensity extraction. So it takes in the center of the cluster an area where it should be clean.
And it's pretty small.
Yes. And also it takes a background around the cluster. And of course you can imagine the higher the concentration of your library, the denser the clusters, and then at some point it becomes really difficult to take the background, and then the signal to noise is going down. Yeah? So this is why you should optimize the concentration. Yeah? If it's too high, it's bad. Of course, if it's too low, it's also bad because you don't get enough data. Right. And the base calling, so we take the intensity data and the cluster location. Output is BCL, which is a binary format. And then we do also the correction for phasing, pre-phasing, and the chastity filtering. So this is how it works. We cluster the signals, and then if it's within this call zone, it can be identified. If it's somewhere here in the middle, it will be rejected. Yeah, so it will be a no call. So these are the call zones. And then the chastity is filtering for basically signal to noise. So you make the quotient of the intensity in one cluster over the sum of the two. So this is the highest intensity divided by the sum of the two highest intensities. So this is illustrated here. So here you're looking at this spot, and this is the center of this cluster. So you take the difference, and this is difference in intensity, and this is the next cluster. And you take also this difference, and you use this to calculate this formula. This is another example. Here it's 0.6. Here it's also 0.6. So here they are equidistant, and then if you have 0.6 divided by the sum, so basically the result is 0.5, yeah, and 1 minus 0.5 is 0.5. So then in this case, and you see here the position is just at the edge, it would be rejected because it's not a very clean signal. Yeah? So it can also be spread from the next cluster. The quality, this is also what you know from MySeq, so this is our quality score, and basically this is what it means. If you have Q10, you have 90% confidence that this call is correct. Q20, 99%, Q30, 99.9%. So most people, they start to use the data if they're better than Q20. Q30, then they're really involved. Yeah. And normally we should also get a significant part of the data that even reach Q40. So this is here you see the cycles, and in the first 5 cycles you find the cluster position, and then once you know all the positions, you can calculate the cluster density. So before you don't really know the cluster density, And then you can also estimate the rate of phasing and pre-phasing, and then you can start base calling and generating BCL files. And then after 25 cycles, you can also do the filtering of the clusters. And then it can start to align to files. So this is then what you can see in the sequence analysis viewer. And then you can calculate an error rate if you compare the result with the file's genome. And also you get the quality score connected to your BCL files. Secondary analysis, we also saw this yesterday in BaseSpace, or BCL to FastQ, that's the other alternative. Yeah. So if you want to use BCL to FastQ, we have a manual for this. Yeah. So it's also you can download it from the same location on our webpage as the software. And this is also interesting. Yeah. So we also have some recommendations about third-party software. About visualization, yeah, so for example, Variant Studio, this Amplicon viewer, to be honest, I think most people don't use, but Variant Studio is quite useful, yeah. So if you are looking, for example, at panels or at clinical exome, then you can use it to annotate and filter your variants. So input is the VCF file and output are classified and filtered variants. So you can filter those variants that are probably disease causing. Tumor normal comparison, RNA-seq, integrative genome viewer, molecular profiler. So these are all applications that you can find in BaseSpace, and then of course you can also use other third-party software. A lot of people, they use IGV or the UCC genome browser. Right, so this was just the overview. Monitoring the quality. You can use a sequence analysis viewer, and I think this is not, you know this already. So this is what you see now. So I think this is pretty self-explanatory. Of course, you know, you get the real information you get after 25 cycles. So these are the data that you need for sequence analysis viewer. Run info, run parameters in the op folder, and potentially also the thumbnails. Yeah, thumbnails are not absolutely necessary, but of course then if you see that there is something strange happening in one cycle, so you usually look at the graph, for example, intensity versus cycle, or you can also look at percentage base call versus cycle, yeah, and then or at the focus versus cycle. And then if you see suddenly some drop or something strange, then you look what cycle it happened, and then you can look at the images before this cycle and afterwards. And this really helps often to understand what happened. And this is also what is interesting then for the support, either Interlabservis or tech support. So this is what it looks like. And the summary, so it really it's the same as on NextSeq. And these are these controls, but most people don't use them. Yeah, only if you spike in during sample preparation. This is quite interesting. This is the statistics of the indices. So here you can see how well the pooling performed. Yeah? So if you have here a lot of variation, you will always have some variation. Yeah? So it's really difficult to make them exactly the same. But if the spread is too much, and here it seems to be quite a lot of spread, then this means you have an uneven distribution of the libraries. So some libraries are over-represented and others are more underrepresented. And of course, this is not optimum, yeah? So then maybe you don't get enough data for this library. So, of course, and if in a mixture you make a mistake with one library quantification, then this will impact automatically all other libraries in the mixture. Yeah. So you have to be really careful to quantify them correctly, and this is why I would really. So, for example, if you pool it. If you quantify by qubit, make at least 2, better 3 measurements, yeah, so that you're really confident that this is correct. Sometimes it can happen that you have only this little droplet, and maybe it's not in the right position, and you get some strange reading.
No, we used 10 nucleases.
Yes. Yeah. Or if there is something with the pipetting, so it's better to have at least two values. Summary. Okay. No. Yeah, we can have a look at later at the machine. And now,
Florian?
Yes.
So I suppose you can download FASTQ files from BaseSpace after they were converted from BCL, right?
Yes.
So basically, I can use BaseSpace to convert the files in FASTQ, and then if there is need, I can use them in third-party software.
Yes, exactly.
Что за BaseSpace? Негативы файлов. Завершился проект, смотрите.
So, BaseSpace OnSite. This is important.
At least in general.
Okay. So, sorry?
I can move the beamer to the right. So
okay. No.
Yeah, okay.
Yeah, so this is what it looks like, and it's connected through a LAN, so a network, to your. So it's a kind of private network, yeah. So not internet, but a private internet. Yeah? So really the idea is for data protection reasons cannot put the data on the internet. You can generate your own network. And it's this parent-child configuration. So inside this network, you can combine several servers and also combine it with some storage. So you should actually do this, yeah. So you should not use the BaseSpace OnSite to store all your data, because and then at some point it needs quite a lot of capacity for the sequence analysis, yeah. So for the data analysis, if you completely fill it up, it will crash at some point, yeah. So you need to think about the data storage. And as I said yesterday, the hard disk only holds one experiment, yeah? So after this it will be full. And also yesterday, remember when we started the experiment, we gave one copy to BaseSpace in the cloud, and one copy, and this is really recommended. It's optional, so you are not forced to do it, but it is highly recommended to have one local copy as a backup. Yeah, in some, for example, apparently the data transfer to the cloud did not. There was also a problem. Yeah, so maybe it's the speed. So at least there is this symbol that says that there is some problem with connection, and it's always good to have a local copy. Okay. So when you have it connected to the storage server, next time you start an experiment, you should give the path of this server when you, to the instrument software. So yeah, these are the difference. So it's connecting to BaseSpace using a local network instead of internet, and all the genomes, so the reference genomes, and the applications and updates, they really have to be downloaded and installed on this server. So now we have installed what we call the core applications, which is RNA-seq and several applications for DNA-seq and enrichment. But we will continuously make updates and also new applications. So you should register. So when you register an account with Illumina, you should get regularly these Illumina notes, yeah, these updates, and please read them, because there we will inform you about any updates and new things. Yeah. So this, then if you know it, then you can get the download. Yeah. But be aware that there will be updates. Okay, so first you have to open BaseSpace OnSite in a browser. So this is really very similar to what we did yesterday. So you also need to log in. And then, yeah, you can have different privileges. So you can. This is up to you how you organize it. So you can use one administrator account that has all privileges, or you can have one administrator and some users that have not all privileges, yeah, for example, to make changes in the software. So this is something you should think how you want to organize it. And yeah, so you can define, so the administrator can define the rules for storage, about user privileges, health checks, planned runs, and so on. Yeah. So probably, I mean, at least for the start, the most easy way is if you use the administrator account. So this is the work cycle. So you have the active storage in your BaseSpace server, and then you have the archive storage in your storage server. And basically, if you want to analyze data in BaseSpace OnSite, you need to unarchive them. But once you have analyzed them, or you don't need them anymore, you should put them back to the archive. Yeah, so in order to make optimum use of the storage capacity you have in the active storage. Yeah, I said already the administrator can manage the user privileges. So these are health checks, so you get also error reports. So this is also something the administrator can see. And then you can also replace power supply module if needed. So it's a modular system. It's fairly easy to exchange different modes. So if you have a problem, we have two guys who are experts in this IT hardware, so if you have a problem there, of course you should tell us, and then they can, usually through an online session, they can really confirm what is the problem, and maybe then they can give you detailed instructions what to do. Yeah. So you can also lock and unlock runs, and we saw it yesterday. Yeah, so we generated a run in the prep tab, and then you have to make it available for sequencing. And basically, yesterday we did this in the cloud, and you can do exactly the same on BaseSpace OnSite. And then for software updates, you can do it through a USB drive. And then you go to the software update page and detect the drive, and then so it's fairly self-explanatory. You will be guided through the procedure. So you can download it from the internet, and then with a USB stick you can upload it. And we have system logs that can help tech support to troubleshoot problems. Yeah, you should not disable the alarm and ignore a warning if it comes. And you should also get the user guide. I can show you where it is, or I can send it to you. But anyway, I mean, since now we were not able to make it running, in January we will bring somebody who will do this. And I think then ideally, I mean, in the optimum case, I don't know if it's possible time-wise, but it would be really nice if you could maybe in the meantime prepare a library for the next experiment, maybe Exxon. But also it can be that since if this run is not good, that we should maybe make another 5x. So this is something we have to discuss. But anyway, it would be good if you could start with library preparation, and then maybe we can start the run together, or just.
But we must discuss it with our boss.
Of course, yes. But I think it would be so if you have a library, then we have more.
We have no library.
Reagents.
We have already library yet.
У нас нет готовой библиотеки. Есть ДНК, который мы можем приготовить в библиотеку. То есть, соответственно, все равно нужно время на процесс. Мы не сможем сразу запустить.
You need to get the sample right.
Alright. They need to prepare the library first.
Yeah. That's what I'm saying. This is exactly what I'm saying. Yes. So sure. Yeah. That's. So I'm just saying that maybe for before the next meeting, it would be good if you could already prepare this library. Yeah. Okay. Good. So I think about software for the moment it's enough. And so maybe later what we can also talk about is the exome sequencing, so the library preparation protocol. If you have any questions there.
It's protocol. Насколько сильно различаются протоколы NextSeq, MySeq?
Is the difference between preparing protocols on MySeq and NextSeq it's very big, or it's the same?
No, the library preparation is the same. So there is not only the only really difference is that when you make the indices, don't take this one 7 or 5. But otherwise, it's the same. But have you done next rapid capture enrichment?
Делали вы
Ну, по-моему, я не уверена, что он даже есть эти наборы.
Yes.
Да,
So.
This is no this kit for enrichment.
Yes. So this is why I thought maybe it's a good idea to speak a little bit about this procedure. And also, and this is what I also want to discuss with Alexander, I think the best would be actually if you could do this course in Heidelberg, where they really in detail and in the lab explain you how to do it. It would really help you to get immediately a good result. Yeah? Otherwise, maybe you need several experiments to optimize it.